Hello, my name is Jennifer Mills. I'm a medical laboratory technician and scientist. Today I'm going to give you a presentation on snake venom and its applications in medicine. A lot of this information comes from the research article called The Chemistry of Snake Venom and its Medicinal Potential. And this was put together by Oliveira and her team at the Department of Chemistry and Biochemistry at the University of Porto, Portugal. Okay, so a little bit of background about this. Venomous snakes use a mixture of enzymes and toxins in order to take down prey. What this does is it immobilizes and allows them to digest it so that they're not moving around the entire time. This same venom also serves as a defense mechanism, one which humans are unfortunately a victim of. A lot of these venomous snakes are fatal if left untreated, if you are bit by one, but still, what we know is that these same toxins have been successfully used in a lot of therapeutic drug administration, even in the modern times. So we obviously know about the anti-venom drugs that we can collect, but we don't know, a lot of information isn't out there about the therapeutic drugs that we do use today that have snake venom. So kind of what this is going to go over is a couple of topics. A lot of this, again, is based on the research of the team in Portugal. The first topic is going to be about the history and kind of how this all works in the first place of extracting venom from these creatures. And then I kind of wanted to talk about the composition of the venom, the chemical properties, what that does to the body, what it does to its prey, things like that. And then we'll go into drugs that have snake venom in them. That are approved by the FDA and used today. And then I kind of wanted to go into two other topics: how we can use therapeutic venom and how important it is to continue the biological research of these creatures in order to benefit both the creatures and human health. So let's get into a little bit of history about therapeutic snake venom. It goes clear back to 60 CE with a tribe called the Siley tribe. They were located in northern Africa. Some of the earliest records about them were written by the Romans, specifically Cassius Dio. He was a Roman historian that lived around 165 to 236 CE. What's unique about this tribe is that they were said to have been bitten so many times by so many different serpents that they were naturally immune to snake venom. So the Roman Emperor Octavian actually sought out this tribe to find a cure for the snake bite that killed Cleopatra. And then they were used many, many other times when the Romans would march across terrains. They were getting bit by snakes and dying. And so they'd actually employ the Siley people to come with them to help treat these bites and give them information on how to prevent the deathly illness associated with them. Another cool little fact is that the Siley were said to test their faithfulness of their wives by allowing a serpent to bite the infant. If the infant died, they were considered illegitimate. Of course, if they lived, they were considered to be a true Silean. So moving on, we've got around 500 CE, we have more records of what they were using snake venom for. And it was usually in topical creams, tinctures, things like that. Sometimes they would ingest the snake venom mixed with other things. And they would do it to treat smallpox, leprosy, fever, wounds, pain, and other ailments. This is just more of an experimental kind of thing that you'd find snake venom in some of these medieval medicines. But it wasn't until 1890 when French scientist Albert Calmet invented antivenom. And so he's kind of a big name, big deal in the antivenom world. He was researching a village in Vietnam when he witnessed 40 people get bitten by a group of cobras after this natural disaster. And it was during that time he was able to do some research into antivenom work. And so that's how he discovered his first antivenom, was by trying to treat those people. So moving on, we have more of our modern era. So 1980, to some of you maybe a long time ago, not so much for me, is kind of when it all started with therapeutic medicine that's actually approved by the FDA using snake venom. They started working on different cardiovascular and pain medicines in about 1980. Especially in the cardiovascular world, and we'll get into that in a little bit. So extraction and use of snake venom is a fairly simple process, especially for making antivenom. And I've included a little video. It's about a minute long if you want to take a look. A really neat, quick way of explaining this. The snake is allowed to bite a container and inject the venom into this container, and then the venom is removed and diluted in a lab, and that is actually what is placed into the horse or sheep, is the diluted form of the venom. So that injected animal gets a chance to make antibodies against this venom. And so then we'll do a blood draw, take the serum from that blood, which is the antibody-rich portion of the blood, and then we'll use that to make the antivenom medication. And the same process happens for the therapeutic drugs. We'll dilute that venom. We may inject it into an animal so that it can break down some of those proteins, and we'll use that sera for medicine. So let's talk about what's inside of snake venom. The most medically significant snakes that they use in research, and the ones that they're extracting this venom for, creating drugs, all that kind of stuff, are those from Elapidae, also called elapids, and Viperidae, vipers. So just if you're not familiar with elapids, they're cobras, adders, mambas, sea snakes, snakes like that. These are all venomous. And obviously the Viperidae are vipers. Now this venom has a lot of different things in it depending on the snake, but for the most part it's made of this 3,9-KDA disulfide-rich peptide, an inhibitor cysteine knot, ICK motif. In other words, it's a chemical that has peptides, proteins, and some enzymes in it that is very toxic to tissue, cardiovascular tissue, skin tissue, the nervous system, those kinds of things. Now, when you're injected with this venom, either by defense or its victim for ingestion, the modes it works is using neurotoxicity that affects the nervous system, or hemotoxicity, that's your vascular system, so your heart, vessels, things like that, and then cytotoxicity, which causes cell death. So the kind of bites where you see that it's swelling and it's really purple and red and painful to the touch, that would be your cell death ones. You see a lot of that with rattlesnakes. So let's kind of get into some of the figures that were presented in Anna Oliveira's work. They decided that the biggest portion of their research was going to go towards discovering and deciding the amounts and types of toxins available in the different medically significant snake species. So they came up with this chart after their research that kind of breaks that down. So you'll see on the left you have the Elapidae types of snakes. That's like the cobras, the hooded snakes. and other similar snakes in that family. And then on the right, they broke down the vipers, the Viperidae family. And so the pie chart, each section of the pie chart determines the type and amount of venom available in that snake. And that's hugely important to scientists when they're trying to research the therapeutic uses of each type of venom so that they can utilize various snake species for the type of research they're trying to perform. And so I'll just kind of quickly go over this chart. On the left with the Lapidae, you have a large portion of that wheel devoted to a dark green color. So if we come down to our legend, we can see that dark green means 3FTX. So if you're a snake enthusiast, and I am not, but if you are, you probably already know what that 3FTX is. That's called the three-finger neurotoxin. Okay, and remember when we talked about neurotoxin, that was anything that affected the central nervous system. And so this kind of snake is going to be the snake they choose to research kind of anything to do with pain relievers, painkillers, stuff like that. So then another large chunk of that wheel you'll see is around 27% of the venom in these type of snakes is that bright orange color. And if we look down, that says PLA2. What's that? Well, that's phospholipase A2. And that's the one that causes cell death. And remember, I told you all the snakes have a bunch of different kind of toxins in them. And so some of them will have a combination of techniques that they use to take down the prey. As you can see, the Lapidae will cause cell death and central nervous system issues. Okay, and so a very large portion of their venom is that PLA2. If we go down, we can kind of talk about the Viperidae. And you can see that the largest part of that wheel is going to be the light green. That's the SVMP type of toxin. That's snake venom metalloproteinase venom. And what that one does, gotta scroll down here. This causes vascular episodes. So the SVMP, and you'll look down, you'll see the SVSP, snake venom serine proteinases. Those are the vascular episodes, heart attacks, red blood cell lysis. Some of them will actually make your vessels weak and collapse and disintegrate, so that causes hemorrhaging. And so that's kind of what the vipers focus on. And again, you'll see they do also have a little bit of that orange stuff. The 20, I think it says 25%, is the PLA2, so that's going to cause a little bit of cell death as well. All right, let's talk about the drugs that we're using. Again, that's post-1980 that utilize snake venom. And bear with me as I try to pronounce some of these. But each one kind of addresses a different thing, and it comes from a different type of snake. So captopril is a drug that they use, very, very, very popular drug that treats hypertension. So what's hypertension? That is high blood pressure. So all the folks out there that have problems with high blood pressure due to plaque, narrowing of the vessels, they have issues with they may have had a heart attack in the past, stroke, things like that. They will prescribe captopril, well, amongst many other types of drugs, but captopril is pretty popular. It's derived from the snake called Bothrops jararaca. The common name for that is the South American pit viper. So another drug is the tirofiban. This one is a little different. It still works in the vascular system, but it inhibits platelet aggregation. platelet aggregation is just platelet clumping. So when you get a cut and you get a scab, that's platelet clumping and aggregation. Unfortunately, that'll happen sometimes inside of your vessels when you don't want it to, causing a blood clot. And so when you get a blood clot, that's going to break loose. Not every time. It may break loose, head to your lungs, brain, cause a stroke that way, or a pulmonary embolism. So tyrofibin is actually derived from Echis carinatus. That's the common name. Common name for this one is saw-scaled viper. Thought that was pretty cool. Another drug that they talked a lot about is eptifibatide. It's another platelet aggregation inhibitor. It just acts a little differently, probably on a different factor. So we have like 13 clotting factors. Some of them work on clotting factor 10, some work on clotting factor 13, and so forth. So that's kind of the difference between tyrofibin and eptifibatide. Eptifibatide is derived from Cistrurus miliarius barborin, Barber's pygmy rattlesnake. So that's rattlesnake venom. So pretty cool, and we use that one pretty regularly. And then our next drug to talk about is batroxabin. This one breaks down fibrins and clots. So the other two, they're just preventing it altogether. This one, you might give this to a patient who's already developed a blood clot. And so you need to break it down, get rid of it before it breaks loose and heads to the lungs or the brain or the heart. And this one, scroll down here. This batroxabin is derived from Bathrops mugini, which is the Brazilian lancehead pit viper. So you can kind of see where these names of these drugs are getting their names from, and they kind of base it off the name of the animal that they used. So moving along, we have hemocoagulase. Okay, the common name for that is reptilase. They use that all the time in our medical lab, in our hematology and coagulation department. Reptilase is they actually even use it in testing, so it's not just something you can give to a person as a therapeutic medicine. You can also use reptilase in your laboratory testing and in the tests that you're performing on the analyzer on blood samples. So this one actually activates clotting factor X, aka 10, which starts a cascade of other coagulation processes. Some people don't have the precursor they need to activate factor 10, and so we can give them reptilase or hemocoagulase to get that going for them. So again, people who have blood clots, their blood tends to clot. Okay, so then our last drug listed here is acobrotoxin. You can probably guess which animal this one comes from. Acobrotoxin has that 3-FTX toxin that we talked about, which is for pain. So this one I think is FDA approved for in America. We don't use it very often. It's more used overseas, especially in China and Asia. It's a very powerful painkiller. So this one is derived from the Naha Atra, Chinese cobra. So you guessed it, it's from a cobra. Moving along here, I did want to talk about a lot of drugs that are in clinical trials right now, because this is fairly new as far as medicine goes. Moving away from, yes, we need anti-venom, but also we're starting to find the therapeutic applications for these venoms, and that's why all these drugs are currently in clinical trials. I wasn't going to list all of them. They are in that article if you want to look at it. But I did want to talk about one of them that I thought was really interesting. There's a team currently using snake venom to target against the SARS-CoV virus. So I thought that was pretty interesting that people would potentially be willing to take a snake venom to target the virus itself. But that's what they're working on. They have found that this specific type of venom is toxic to the COVID virus and SARS as well. So this one that they're working with is actually derived from Bungarus fasciatus, which is the banded krait. And it's a combination of a couple of venoms, also from the Vipera renardi, which is the steppe viper, and Vipera nikolowski, which is Nikolowski's viper. So they're using krait and viper venom in this research study. So let's just talk about, how you can use snake venom. I know we kind of talked about those drugs and broke them down and what they do, but overall, you're going to want to use them for to treat high blood pressure, blood clots, and chronic pain. And again, that viper venom is better for blood pressure. We went over that in our chart. Rattlesnake venom is better for people who develop blood clots. And to prevent platelets from sticking together. And the mamba venom, and that area of species is better for chronic pain. So any of the adders and those types of snakes would be better for because of their neurotoxin venom. So something on the horizon is cancer treatment. So there's a lot of early clinical studies about how we can use snake venom to treat cancer. We have the cytotoxic venom that eats tissues, so we started thinking maybe it would eat tumor or cancer cells as well. And it does. It interacts with the lipoproteins in the plasma membrane of cells, which, in layman's terms, causes the cell to shrink. And that's definitely what you want in cancer, want to happen for cancer treatment. So our best snakes for that would be the elapid snakes, because they have proven effective against melanoma and sarcoma types of cancers. Those are the lumps in clinical trials. So why is this all important to research snakes, their habitat, their health, all that kind of stuff? Because field research gathers information about these species, we're learning new species through this research. We can keep the populations healthy. We're able to categorize and discover new things about these animals. We can protect their habitat. And it's allowing them to create antivenoms for bite victims. So we don't want anyone to die from these snakes, so we are regularly collecting that. If we lose all these snakes, we won't be able to do that. And healthy snake populations allow us to harness venom for drug discovery, which is what this whole presentation has been about. Another thing that they're finding, and I didn't choose this type of research, but it would be something cool to do in the future, is snakes are the model organism for evolutionary studies. So geneticists and that kind of biological study really rely on snakes to tell them the story of evolution. And a lot of other molecular studies can be done with snakes to learn more about these organisms. So to wrap it all up, the conclusion of this is that researching and protecting venomous snakes, we help at the same time contribute to human health. We're having a lot of medical breakthroughs, of course, the antivenom, and even more research because of these animals. And in order to have these breakthroughs, we have to provide healthy habitat, track these snakes, and do lots of research to make medical discoveries. And thank you very much for watching my presentation. I hope you learned something, and I hope it wasn't too long to watch. And thank you very much.